All right, so this is the AHL version of the Chapter 17. So this is some additional stuff that we learned about in class that the AHL students only need to know. And the idea here, the essential idea is that information stored as the DNA gets copied into messenger RNA, so we're going to expand upon that a little bit. And the information transferred from the DNA to the messenger RNA is transferred into the amino acid sequence. And again, we're going to expand a little bit upon that. So very similar to some of the stuff that we talked about in class, talked about with the just the regular core information. So this is just a little bit more. So from a TOK standpoint, I need you guys to look at the question, the nature versus nurture debate concerning the relative importance of an individual's innate qualities versus those acquired through experience is still under discussion. Is it important for science to attempt to answer this question? So take a second, write that down, write down your response, and then we'll discuss it in class, okay? All right, so the main questions here, just kind of like we talked about earlier, the information content in the DNA is specific sequences of nucleotides along the DNA strands. Of course, the order of the A's, T's, C's, and G's that we talked about. So how does this information determine the organism's appearance, and how does the information in the DNA sequence, how is that translated into the cell into a specific trait? Okay, so of course transcription and translation, these are things we talk about, going from the information on the DNA, the gene, splicing out all of the introns, linking together the exons, and then getting a functional piece of messenger RNA created in the case of a eukaryotic cell, or in the case of a prokaryotic cell, just simply making the messenger RNA, and then using that message on that messenger RNA to turn it into protein at the ribosome. Okay, and we discussed some of the things in a bacterial cell, how it can occur at the same time. Okay. So we talked about transcription and translation is not separated because there's no nucleus. So it occurs at the same time, and then in a eukaryotic cell like we've got right here, transcription occurs in the nucleus. You get the messenger RNA processing, you get rid of the exons, get rid of the introns, link together the exons, and then they modify the ends a little bit, and then get that out into the cytoplasm, where it's going to link up with a ribosome, either free or bound, and become protein. And we discussed, of course, this template strand, the non-coding, the antisense, giving rise to this messenger RNA molecule, which becomes protein at the ribosome. Okay, so we went through those details in the last one. And a little bit of what we talked about was that RNA was used because it was a way to protect kind of that master copy, so to speak. So if anything happens to RNA, we can always make more because we have the DNA, whereas if we were working from the DNA and something happened, then that would create a problem for us. So we can also manufacture many copies of the messenger RNA at one time, and then from that we can establish still kind of almost geometrically more protein, because we can have multiple ribosomes that can be transcribed, or that can do the transcribing of the gene, or translating of the gene, into the protein through that structure called the polyribosome, which we were talking about. Okay, and we watched a movie on that. We discussed a little bit in class and those sorts of things. Okay, and then recall, of course, the main difference. We said the main difference between the prokaryotic and eukaryotic cells is what the processes kind of at the heart are the same, but they occur at the same time in a prokaryote and different times, of course, in the eukaryotic cell. We looked at this chart here. We were talking about the start codon, the 3 stop codons, and then the 61 other codons that code for the amino acid. And we used that in a couple of examples. And then we also talked a little bit about how that genetic code is redundant. In other words, there's more than one triplet that can code for the same amino acid. Like, for instance, if we look right here at this phenylalanine, we can see that there's 2 that code for phenylalanine here. With leucine, there's a lot of redundancy. In other words, if you have, in this case, the first 2 are U's, you put in an A or a G here, you're going to still get leucine. Same here, if you have Cu as the first 2, any other base pair is going to give you the leucine, and there's a lot of other examples as well. So that's what we mean when we say that this code here is redundant. Okay, and then really only one triplet, though. Only one triplet is going to code for any amino acid. So we're not going to have, in other words, if we back up here, UUU is always going to code for phenylalanine. It's never going to code for any of these other amino acids. All right. And then the reading frame, we discussed a little bit about the reading frame. That's the order of the A's, T's, C's, and G's, or on the messenger RNA, rather, the A's, U's, C's, and G's. That reading frame is important, because any error, any addition, any deletion, any kind of inversion, or anything like that that alters the reading frame is going to give us what we call gibberish, which is basically nonsense. It's a protein that really isn't going to do anything. Okay. So Transcription, this is where the messenger RNA molecule is going to get made from that template strand, and it's always going to be read 3 to 5, so that messenger RNA, of course, can be built 5 to 3. Those codons then are read 5 to 3 by that ribosome. We went over some examples of this in class, and this is where we get this protein that's starting to take shape. Okay, and that messenger RNA, this is what's going to carry that information, of course, again to that ribosome. The polymerase, the RNA polymerase, is going to pry apart that DNA to give rise to that messenger RNA. Again, this is just kind of some reviewing of some of the things that we talked about. And we talked about this RNA polymerase 2. This is what it's going to do, is it's going to encounter a promoter. And we have over here, we have the promoter, this Tata box, this whole idea right there. This is where this entire kind of complex that you see down in this region here is going to start to form. So you get all of these things with the TFs on them. These are the transcription factors. There's a bunch of those that are involved. And then these things all kind of jump on. And then within this entire structure here is where this RNA molecule is going to start to get manufactured. So you need all of these things to kind of encounter this sequence for this entire thing to kind of get started. And then once it reaches that terminator sequence, of course, that's where we're going to kind of end this process. Okay. And prokaryotes have one type of RNA polymerase, and this is going to synthesize the messenger RNA, and then there's other types that make other types of RNA, like the ribosomal RNA and the transfer RNA and things like that. And then eukaryotes, they have 3 types of this RNA polymerase in their nucleus. And RNA polymerase 2, this is the one that's important for synthesizing the messenger RNA that we've been talking about. Okay, and then we talked about the idea of a promoter that's going to get this whole process of transcription started at the gene, and this is where the RNA polymerase molecule is going to attach. We looked at a video that shows how this whole thing is kind of working. All of these transcription factors start to bind on the enhancer regions. We're going to basically begin to express this gene as the DNA starts to wrap around and kind of get involved with this entire process. Okay, and then when we reach that terminator sequence, that's where DNA replication, or the messenger RNA replication, I should say, is going to stop. Okay, and these promoters now, just expanding on this a little bit, this is usually a few dozen nucleotides upstream from that transcription start point. So this promoter is going to get the process going. We said, of course, this includes the TATA or the TATA box in the eukaryotic cells, and these promoters are important for the binding of these RNA polymerases. And those transcription factors, these are the proteins that are going to kind of interact with and bind to the promoter region. That's what's going to enable this RNA polymerase to kind of do its work. So again, in this other schematic that we've got right here, you can see where these transcription factors are starting to bind, and that's what's going to facilitate the attachment of this RNA molecule to this. And then within the structure now, again, we said that's where the DNA is going to be kind of pried apart, and that's where this transcription is going to begin to take place, adding to that 3' end. Okay, and then we looked at something very similar in class to this molecule here. We said that as the DNA is brought in, it gets spread apart. This, of course, if I change the colors, it might be a little bit easier to see. This is the 3' end, because this is where we're adding to the 3' end. This, of course, is that 5' end. And then as this RNA polymerase kind of moves in this direction, or as the DNA is moving in that direction, that's where we're going to slide along. These nucleotide or ribonucleotides, these things are going to be brought in kind of to this funnel, and if the correct one is there, then we're going to add it on to this growing chain and just continue to slide along until we reach that terminator sequence where this process is going to end. In prokaryotic cells, it's important to know that the promoter is associated with the sigma factor rather than these transcription factors that eukaryotes are kind of interacting with to express this gene. The sigma factor is essentially going to do the same thing for our purposes, the same thing that the transcription factors are. That's going to help this, synthesis of the messenger RNA molecule. Okay, so those things are, the sigma factor and the transcription factors are very similar to one another. Okay, and then once they bind, of course, this is where you get this transcription initiation complex, which is what this entire thing right down here is called, and that's where we're going to start to begin transcription. And as it moves along, it uncoils the DNA, of course, going to synthesize that messenger RNA. And It's important to know now that numerous RNA polymerases can join on to that DNA sequence and synthesize large amounts of protein really and messenger RNA at the same time. And this is what we've discussed in class, enables the cell to make large amounts of protein in a very short period of time. And you can see these structures here that are called Christmas trees, because if you kind of stand them up on end, they kind of represent like a pine tree. This is where the process is beginning. Okay, and I can switch my color right here. So this is where this process is beginning, and then this down here is where it's ending, because you can see these things as we move along here are getting progressively longer, okay, compared to say this end where they're a lot shorter. And you can see this is multiple structures that we see here. This is where we're transcribing, in this case, two particular types of genes. All right, so if we were to look at this in a bacterial cell, you could see how this is occurring. If you were to look at this, hopefully you would be able to know that it's a bacterial cell, because this is the RNA polymerase molecule. This blue line here, this is the DNA. And then what we're doing is this red here is the messenger RNA. And these purple lines that we see right here, these are the proteins. So in this case, we've got transcription and translation that are occurring at the same time. And because there's no nucleus, there's no RNA processing, we know that this process is occurring in a prokaryotic cell. Okay, and then once we get to the end of this DNA sequence of transcription, this is where the RNA polymerase molecule is going to detach from the DNA, and that's that terminator sequence. And this makes the messenger RNA immediately available for use. Of course, in prokaryotes, which we just saw, it might need to be processed a little bit in a eukaryotic cell. Okay, and then there's a signal sequence now in the eukaryotic cells, that once we kind of get to that spot, this is where it's going to cleave off this RNA molecule from this growing chain, and then the RNA polymerase is going to kind of continue off. It's not very well understood how the process ends, and then we end up with this RNA molecule that's really not fully ready to use. Okay, so we'll pick up with the RNA modification here in the second one. Hopefully that part one here reviewed some of the stuff that we've discussed in class and gave you a little bit of new information. So we'll talk to you soon.